Hey guys, and welcome to Video Notes Part 2, and it's about DNA replication. So, 10.4 we start off talking about how DNA replication depends on specific base pairing. So remember how we already talked about how A pairs with T and C pairs with G. So, DNA replication will follow a semi-conservative model. So this means, okay, so we have two DNA strands because the original DNA strand will separate and we'll have two left over. So each strand is used as a pattern to produce a complementary strand using that specific base pairing that we talked about. So then each new DNA helix that's formed is semi-conservative because it has one old parental strand with one new made strand. So let's take a look. So our blue here is our parental strand. It's our original DNA. It splits apart and opens. And that gray there is that new DNA molecule. And that is based off of the ATCG base pairing rules. So here's our parental molecule of DNA, that blue there. It splits apart, and based on the nucleotides on that parental strand, that new daughter strand is formed. And when we have the end result, we have two identical DNA. Remember how all the DNA in our cells is identical. And we have one that's completely new, a strand that's completely new, and then we have another strand, that gray strand there, that is new, and then that blue strand that is old. So here's our parental part and our new part of DNA. And the end result is two DNA that are completely identical. So in 10.5, DNA replication proceeds in two directions at many sites simultaneously. Now this kind of gets a little bit confusing. So DNA replication begins at a site called the origin. Okay, this origin kind of looks like a bubble inside DNA. So it kind of pops open, forms a bubble, and then replication begins. So this replication is going to proceed in both directions, to the right and to the left, because we want to do it as fast as possible. We want to replicate our DNA fairly quickly. So when you get a cut, it heals extremely fast. Replication ends when the products from those bubbles come together. So when those bubbles merge, that's when our replication stops and our new DNA is formed. So let's take a look. So here's our origin of replication. Okay, that's our bubble. You see how we have many different bubbles? That's really important because that helps DNA replicate much, much faster. So here's our bubble. And it's going to replicate in both directions. Remember, both directions. So when these different bubbles meet each other, that's when our replication will stop. And let's take a look at our end result. See, the blue is our old strand, the gray is our new, and we have two that are produced. And it's called semi-conservative, half new, half old. So DNA replication occurs in the 5' to 3' direction. So we're not going to get into the nitty-gritty details because that's no fun for this chapter. We need to take it step by step so we can understand it. And too much at once will just be confusing. So DNA replication occurs in that 5' to 3' direction. Replication is continuous in the 3 to 5', but discontinuous means start, stop, in the 5' to 3'. And it's going to form short segments. So let's take a look really quick. So DNA runs from the 5' to 3' end. And what that has to do with is that sugar backbone. Those carbons in the sugar are labeled 1, 2, 3, 4, 5. And you won't need to know that. You just need to know that it runs in the 5' to 3' direction. So DNA replication proceeds in two different directions and many different origins. So we just took a look at that. So we have many different bubbles that are produced, and that replication will run in both directions, right and left. So the proteins involved in DNA include something called DNA polymerase. So that adds nucleotides to that new DNA strand. So we talked about A and T, G and C. So that DNA polymerase is the protein that adds those nucleotides to the new strand. DNA ligase is going to join those small fragments we talked about, how it starts, stop, start, stop. That's going to join those fragments and make it a continuous strand, because we don't want our DNA to have starts and stops in it. We want it to all be continuous. So let's take a look at this picture here. So here's our DNA polymerase, and that's what adds those nucleotides to that new daughter strand. You see how whenever you see gray, think of new. So that DNA polymerase is adding those nucleotides to that new strand. Down here it shows DNA ligase. That's what's going to join those fragments together so that we have one continuous strand. So you see how the 5 to 3 is synthesized continuously. There's no starts and stops. However, you look down here, you see a bunch of starts and stops. It's synthesized discontinuously. So it's really important that that DNA ligase can come in and join those fragments together. That's really, really important. So the flow of genetic information from DNA to RNA to protein. So this is the basic central dogma of biology. Everything is made of DNA. So what we do is we take that DNA that makes up our genotype. So remember how when we think of genotype, we think of those letters. Well, those letters are that part of DNA. And how it's expressed is through proteins. So our DNA is transcribed into RNA, and then that RNA is made into proteins. That proteins is what's going to provide the way we look. It's going to provide our phenotype. So a gene is a sequence of DNA that directs the synthesis of a specific protein. So we talked about how those sequences in DNA are genes, and we have a lot of genes on our chromosomes. So what those genes do is it makes specific proteins. So first we go DNA to RNA. So DNA is unwound so a strand of RNA can be formed. Then that RNA will leave the nucleus and go outside into the cytoplasm where it can be translated into a specific protein. And the presence and action of those proteins we make in our body is what determines our phenotype. So genetic information is written into codons. Those codons are a set of 3 nucleotides, such as A, C, T. And then those codons, those set of 3 nucleotides are going to be translated into an amino acid. So remember how proteins are broken down into amino acid sequences, and then so a long chain of amino acids can be considered one protein. So the sequence of nucleotides in that DNA is going to be what eventually constructs the proteins in our body. So protein is going to require a conversion of nucleotide sequences into an amino acid sequence. So what happens is transcription is going to rewrite that DNA code into RNA. So it's the same nucleotide language. So what it means is that DNA is unwound, and from that DNA an RNA can be made. So it's the same language. We're using nucleotides. So each word, so each set of 3 letters is considered a codon. Okay, so that codon has 3 nucleotides, like A, C, T. Then translation involves switching into a different language. Okay, so translation, we're taking the RNA and goes into the cytoplasm, and we're making it into a specific amino acid. And then each amino acid is specified by a codon. So each triplet pair, like ACT, codes for a certain amino acid. So then we're going to make a bunch of amino acids that eventually form a protein. So let's take a look at a picture. So we have our DNA in the nucleus. It's transcribed into an RNA molecule using the same language, such as ACT. And then it leaves and goes into the cytoplasm and is translated into amino acids. So each one of those little tiny purple molecules right here would be an amino acid, and then those amino acids will combine together to form one long protein. Then let's take a look at the overall picture. So we have a bunch of genes on our DNA. Those genes are copied, transcribed into RNA. Now this is something kind of new. Our A's in DNA will turn to U's in RNA. Okay, and we'll talk a little bit more about that in class, because it's kind of something that you just have to remember. So A's go to U's, but our G's will still pair with C's, and so on. So each 3 is a codon. Remember, each 3 nucleotides is called a codon. And each one of those codons, each one of that triplet, codes for an amino acid. And those amino acids combine to form a really long polypeptide protein. So here, take a look at this again. Try to pause the video and explain it to yourself how I just explained it to you. So 10.8, the genetic code is the basis of life. So depending on the characteristics of that genetic code, those 3 nucleotides will specify 1 amino acid. You don't really need to know that 61 codons correspond to amino acids, but it's kind of interesting. So AUG will code for the start of transcription. So what's kind of cool is we have a triplet code, a codon, that starts transcription, and we also have some that stop. So how else is the body going to know when to start and when to stop transcribing? So that's really important. So it's kind of redundant in the sense that more than one codon will code for an amino acid. So maybe ATU and GAU are both going to code for a specific amino acid. However, each codon for one amino acid does not code for any other. And it doesn't contain spacers or punctuation. It's just adjacent with each other, and there's no gaps in any of the letters. And it's pretty much universal, which is really, really interesting because everything has DNA. So it's really cool if you think about evolution. Everything has DNA, and those specific codons determine that protein in almost all organisms. So just take a look at this really quick. It's not going to make a whole lot of sense until we talk about it in class. But we think of that triplet code. We go to the first base to say the first base is C, the second base is A, and the third base is G. So A, C, G code for that protein THR. So I know that's a little rough, but look at it at your book, look at how it explains it, and then we'll talk about it more in class. So I promised you guys that you would get to meet Glock. So let's take a look at my husky. Let's meet the kids at home. Say hi. Say hi. Say hi. Say hi, Rube.